In this series of videos we explore the depths of sequence comparison. The illustrations and the main informations for this series are based on the slides of the lecture on Foundations of Sequence Analysis by Stefan Kurtz, Center for Bioinformatics, Universitaet Hamburg, 2022. Whether you're new to the field of bioinformatics or an experienced researcher, this video will introduce the basic idea of sequence comparison and its importance in understanding the biological world, the edit operations and alignments. So let's get started.
I heard about sequences and sequence comparison in the context of bioinformatics. But I think I don't get it.
In bioinformatics we often look at DNA sequences, which is a string of nucleotide bases, A, T, C, G. Sequence comparison is a fundamental concept in bioinformatics that involves aligning two sequences of DNA, RNA, or protein to identify regions of similarity or difference between them. With the rapid growth of genomic and proteomic data, global sequence comparison has become an essential tool for understanding the structure and function of biological molecules. By comparing sequences from different organisms or different genes within the same organism, scientists can gain insights into the evolutionary relationships between species, the mechanisms of disease, and the basic biochemical processes that underlie all living organisms.
Thank you, but what is the difference between local and global sequence comparison?
The main difference between these two methods lies in the way in which they align the sequences. In global sequence comparison, the entire length of the sequences is taken into account during the alignment process. This means that the sequences are compared from start to end, and gaps can be inserted at the beginning or end of the sequences to achieve the best alignment. Global sequence comparison is useful when the two sequences being compared share significant similarity across their entire length. In contrast, local sequence comparison only focuses on the regions of the sequences that share similarity. In other words, only a portion of the sequences is aligned to one another, and gaps can be inserted within the sequence to achieve the best alignment. Local sequence comparison is useful when the sequences being compared are similar in some regions, but differ significantly in others.
That's cool. And how do we compare two sequences?
Usually, the costs for transforming of the first sequence U to the second sequence V serves as measurement of similarity between the two sequences. Here we need to introduce the edit operations. Edit operations describe insertions, deletions and replacements of characters inside a sequence. An edit operation is a pair alpha and beta, where alpha and beta are elements from an alphabet, for example, A, T, C, G, or the empty string epsilon. But alpha and beta cannot both be epsilon. Such an edit operation is usually written with an arrow from alpha to beta. Let's take a closer look at the three types of edit operations. A deletion deletes a character A. An insertion inserts a character B. And a replacement replaces a character A with a character B. Moreover, insertions and deletions are commonly referred as indels. For example, in the case of DNA sequences, AG would be a replacement from adenine with guanine, or epsilon G would be the insertion of guanine.
Interesting. Is there a way to summarize the transformation of one sequence to another sequence?
Of course. Such a transformation is summarized inside an alignment. Alignments are a sequence of edit operations alpha 1 beta 1 up to alpha H beta H. An alignment of U and V is a sequence of edit operations, where U is equal to the sequence alpha 1 to alpha H and V is equal to the sequence beta 1 to beta H. For a compact alignment representation, we write the sequence alpha 1 to alpha H over the sequence beta 1 to beta H. Take the note that an alignment of two sequences U and V is not unique. Here is an example of some alignments for the sequences U equal GATH and V equal GCDHT.
I believe that I now have a comprehensive understanding of sequence comparison, edit operations, and alignments.
Thanks for watching this video. Feel free to ask questions in the comments section and we'll see you in the next video about sequence comparison.